Hello, friend. Welcome to Science for Sleep. Whether you have been here many nights before or are finding this channel for the first time, this is a quiet place to slow down, learn something gentle, and let the day gradually fall away. Tonight, we are looking at one of the most familiar small mysteries of summer. Two people can sit beside each other on the same porch, at the same picnic table, or near the same patch of grass, and have very different experiences with mosquitoes. One person may barely notice them. The other may spend the evening brushing mosquitoes from their arms and waking the next morning with several itchy bumps. That difference is not always imagination. Laboratory experiments have shown that mosquitoes can be much more attracted to the scent of one person than another, and those preferences can remain surprisingly stable over time. The reason is not that one person has sweeter blood. A mosquito usually begins choosing a target before it has touched the skin, which means it cannot know how anyone's blood tastes. Instead, a female mosquito builds a target from several clues at once. She can respond to carbon dioxide in breath, body odor, warmth, moisture, visual contrast, and other chemical signals released from the skin. Those clues overlap, and their strength is slightly different for every person. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. One of the most important clues seems to be human body odor, although not simply whether a person smells sweaty to another human. Our skin releases a large mixture of chemicals that mosquitoes can detect in amounts far too small for us to notice. Some of those chemicals come directly from skin oils and sweat. Others are changed by the bacteria and other tiny organisms that normally live on the skin. This means that every person is surrounded by a slightly different chemical signature. In 2022, researchers studying the mosquito Aedes aegypti tested this idea by collecting human skin odor on nylon sleeves worn on the forearms. The mosquitoes were then allowed to choose between odor samples from different people. The differences were striking. Certain participants attracted far more mosquitoes than others, and people who ranked as highly attractive tended to remain highly attractive when they were tested again over a period of several years. Chemical analysis found another pattern. The most attractive participants tended to have larger amounts of several carboxylic acids on their skin. Carboxylic acids are a broad family of common organic chemicals. Some are produced through normal skin chemistry, especially from oily substances made by glands in the skin. They are only part of the scent mixture, but they appear to help explain why some people consistently stand out to Aedes aegypti. That still does not mean there is one chemical that turns someone into a mosquito magnet. Mosquito attraction is a combined sensory process. A person's metabolism affects how much carbon dioxide they produce. Activity can change breathing rate, body temperature, and sweating. Skin microbes help shape body odor. Inherited traits may influence parts of skin chemistry and physiology. Even the mosquito species matters, because thousands of mosquito species exist, and they do not all prefer the same animals or search for hosts in exactly the same way. There is also one important detail hidden inside the word bite. Only female mosquitoes take blood meals. Male mosquitoes do not drink blood from people or other vertebrates. Both males and females can feed on plant sugars, including nectar, which supplies energy for flying and ordinary activity. For females of many species, blood has a different purpose. It provides nutrients needed to develop eggs. So the mosquito approaching an arm is usually not searching for her everyday source of energy. She is responding to a reproductive need, using a sensory system shaped to locate a suitable animal host. That simple summer encounter connects human chemistry with mosquito senses, reproduction, evolution, ecology, and, for a smaller group of species, the spread of disease. But the first choice happens before any of those later consequences. Somehow, a mosquito moving through open air must discover that a human body is nearby. The first clue leaves your body every few seconds, carried into the air with each breath you breathe out. Inside the body, that signal begins as a normal product of staying alive. Every cell needs energy, and much of that energy comes from breaking down nutrients with oxygen. One of the waste products of this process is carbon dioxide. The gas moves from working tissues into the bloodstream, travels back toward the heart, and is carried to the lungs. There, it crosses into tiny air sacs called alveoli and leaves the body during exhalation. The difference between that breath and the surrounding air is surprisingly large. Ordinary outdoor air contains only about 0.04% carbon dioxide. Air leaving the lungs contains roughly 4%. That makes an exhaled breath about 100 times richer in carbon dioxide than the atmosphere around it. To a mosquito searching for an animal, that difference can be useful information. Female mosquitoes have sensory nerve cells that respond to carbon dioxide. In Aedes aegypti, the species responsible for spreading diseases such as dengue and yellow fever, part of this sensing system is located on structures near the mouth called maxillary palps. These small paired organs contain specialized receptors able to respond when carbon dioxide rises above the background level. The word receptor simply means a biological structure that reacts to a particular kind of signal. When a female Aedes aegypti encounters a suitable rise in carbon dioxide, her behavior can change. She becomes more active and begins searching more strongly for the source. Other clues, including odor and visual contrast, can also become more useful once this searching state has been triggered. The gas does not form a neat, invisible line between a person and the mosquito. Outdoor air is constantly moving. A breeze stretches an exhaled cloud, while turbulence breaks it into patches containing different concentrations. A mosquito flying through that air may encounter a brief pocket of stronger carbon dioxide, leave it again, and then cross another patch a moment later. By responding to these changing pockets along with the direction of moving air, the insect can gradually orient toward the area where the gas is being produced. Distance varies greatly with wind, mosquito species, temperature, and the amount of carbon dioxide being released. For that reason, there is no single detection distance that applies to every mosquito in every setting. Under useful conditions, though, carbon dioxide can begin influencing host seeking from several meters away. This also helps explain why the amount a person produces can matter. A larger adult generally has more metabolically active tissue than a small child, and usually releases more carbon dioxide in absolute terms. Activity can raise the signal further. During exercise, muscles consume energy more quickly. The body produces additional carbon dioxide, and breathing becomes faster and deeper to remove it. Someone who has just been running may therefore produce a stronger breathing signal than the same person sitting quietly. Pregnancy can alter several of these signals as well. As pregnancy progresses, metabolism increases to support the developing baby. Breathing patterns change, blood flow rises, and the body can produce more heat. Studies involving some malaria-carrying mosquitoes have found increased attraction toward pregnant women, although carbon dioxide is only one part of that larger physiological change. Even at rest, every person continues releasing carbon dioxide with every breath. That gives a searching mosquito a dependable sign that a living, breathing animal is somewhere nearby. Its usefulness also creates a major limitation. A person is far from the only creature producing it. Dogs exhale carbon dioxide. So do deer, birds, horses, and countless other animals that mosquitoes may encounter. A carbon dioxide plume can therefore announce the presence of a possible host without clearly identifying what kind of host produced it. As the mosquito moves closer, broader evidence of life has to be narrowed into something much more specific. Once a female mosquito has entered an area carrying signs of a breathing animal, finding the actual body becomes a more precise task. Carbon dioxide may have brought her closer, yet the gas is constantly shifting through moving air. She needs additional information to turn that broad chemical signal into a location. Vision begins to help. Aedes aegypti does not see the world with the sharp detail available to human eyes. Its compound eyes are built from many small visual units, and they are especially useful for detecting contrast, movement, and differences in light. Experiments have shown that once carbon dioxide activates host-seeking behavior, these mosquitoes become more interested in visible objects around them. Dark shapes can become especially noticeable against a lighter background. Researchers have also found responses to certain wavelengths of visible light associated with the colors reflected by human skin. This does not mean the mosquito sees a person as a detailed face, arm, or leg. Visual information gives it another way to organize the space around the source of the breath it has already detected. As the distance closes, warmth adds another layer. Human skin is usually cooler than the body's internal temperature of about 37 degrees Celsius. Exposed skin often sits somewhere in the mid-30s, depending on the room or outdoor temperature, blood flow, clothing, and the part of the body being measured. That warmth can reach a mosquito in several ways. Direct contact transfers heat when the insect touches the skin. Moving air can carry warmth away from the body through convection. Thermal infrared radiation also leaves warm surfaces continuously. Infrared is a form of electromagnetic radiation with wavelengths longer than visible red light. Humans cannot see it with ordinary vision, although thermal cameras can turn differences in infrared energy into visible images. For many years, researchers knew that mosquitoes responded to warm objects at close range. A study published in the journal Nature in 2024 added a more surprising detail. Researchers working with female Aedes aegypti found that thermal infrared from a surface warm to about 34 degrees Celsius could increase host-seeking behavior. That temperature was chosen because it closely matches warm human skin. The effect became especially strong when infrared information was combined with other host signals, including carbon dioxide and human odor. Under those experimental conditions, thermal infrared on its own produced a much weaker response. This fits the larger pattern of mosquito sensing. Each signal becomes more useful when it agrees with the others. The same study found evidence that a heat-sensitive protein called TRPA1 plays an important part in detecting this thermal information. A protein like TRPA1 acts as an ion channel. When conditions around the sensory cell change, the channel can help trigger electrical activity in the nervous system. Researchers traced an important part of this infrared response to sensory neurons near the tips of the mosquito's antennae. They also estimated that thermal infrared could provide useful directional information from around 70 centimeters away under their laboratory conditions. That is roughly the distance from a person's shoulder to the end of an outstretched hand. Air movement, temperature, humidity, and the surrounding environment can change how useful any particular cue becomes outdoors. A mosquito therefore approaches a host by combining several imperfect pieces of information. Carbon dioxide can activate the search from farther away. Visual contrast can help locate possible objects in the environment. Body-associated warmth becomes increasingly useful as the insect draws nearer. Together, those senses can turn the general presence of a breathing animal into the much clearer location of a warm body. Yet a warm body still does not explain why two people sitting close together can receive very different levels of attention. That difference becomes much easier to understand when the mosquito reaches the cloud of chemicals constantly leaving human skin. Near the skin, the mosquito enters a much richer chemical environment. Human skin is constantly releasing small amounts of material into the air. Most of it is far too faint for another person to notice. For a mosquito, some of those molecules carry useful information. What we casually call body odor is actually a mixture of many different chemicals. Some come from sweat. Others come from skin oils, dead skin cells, or substances produced by glands beneath the surface. One important source is sebum. Sebum is an oily mixture released by sebaceous glands, which are found in large numbers around areas such as the face, scalp, chest, and upper back. It helps keep skin flexible and reduces water loss. Chemically, it contains fats and other oily compounds that can later be broken apart or changed at the skin surface. Sweat adds a different set of ingredients. Human sweat is mostly water, along with dissolved salts such as sodium and chloride. It also contains smaller amounts of organic compounds produced by normal metabolism. Different sweat glands contribute somewhat different mixtures. Eccrine glands are spread across much of the body and play a major role in cooling. Apocrine glands are concentrated in places such as the armpits and become active around puberty. Together with skin oils and other secretions, these materials create the chemical starting point for human scent. Researchers have identified hundreds of volatile and semi-volatile compounds associated with human skin. A volatile compound can evaporate easily enough to enter the surrounding air. A semi-volatile compound evaporates more slowly, although some still reaches the air around the body. These molecules belong to many chemical families. Carboxylic acids are one important group. Human skin also gives off aldehydes and ketones, along with various alcohols and many other odor-producing substances. Their concentrations can be extremely small. A mosquito does not need the air around a person to smell strong by human standards. Its sensory system is built to detect particular molecules at low concentrations. Much of that chemical sampling happens through the antennae. A mosquito antenna is covered with tiny sensory structures called sensilla. Inside many sensilla are receptor neurons. These are nerve cells equipped to react when certain airborne chemicals reach them. Two major groups of mosquito chemical sensors are called odorant receptors and ionotropic receptors. Both help the nervous system separate useful chemical signals from the much larger mixture of molecules present in ordinary air. Different receptors respond best to different chemicals. The mosquito can therefore receive information from several parts of an odor mixture at once. That becomes especially useful when the insect is trying to distinguish a preferred host from everything else nearby. The receptor system also varies among mosquito species. Aedes aegypti has evolved a strong preference for humans in many parts of its range. Other mosquitoes regularly feed on birds, livestock, wild mammals, or several kinds of animals. Those differences are reflected partly in which chemical signals their sensory systems are best prepared to notice. Human scent also changes across the body. The chemical mixture near a forearm differs from the mixture around a foot or scalp. Temperature, sweat production, skin oil, recent activity, clothing, humidity, and the local condition of the skin can all shift the amounts of individual compounds being released. Even the same person does not carry one perfectly fixed scent from morning to night. Still, some parts of a person's chemical profile can remain surprisingly consistent. That helps explain why controlled mosquito experiments can find stable differences between people even when the tests are repeated many times. A mosquito approaching skin is therefore sampling a moving chemical mixture whose overall pattern can still carry a recognizable biological signature. And part of that signature comes from organisms living directly on the skin, where they quietly change human secretions into entirely new odor molecules. At the surface of the skin, human chemistry becomes mixed with the activity of entire communities of microorganisms. Healthy skin naturally carries bacteria, fungi, and other microscopic organisms. Together, these communities are called the skin microbiome. The word microbiome refers to the collection of microorganisms sharing a particular environment. That environment changes across the body. The forehead and upper back tend to be relatively oily because they contain many sebaceous glands. The armpits and spaces between the toes are warmer and more humid. Areas such as the forearm are usually drier. Each setting favors somewhat different microbes. Among the common bacterial groups found on human skin are Staphylococcus, Corynebacterium, and Cutibacterium. Their proportions vary from one person to another, and from one part of the same person's body to another. That variation matters because bacteria can change the chemicals released onto the skin. Fresh sweat provides a useful example. Sweat leaving an eccrine gland is mostly water, with salts and smaller amounts of compounds such as amino acids. By itself, freshly produced sweat often has relatively little odor. Once it remains on the skin, bacteria begin using some of those compounds as nutrients. Their metabolism breaks larger substances into smaller molecules that can enter the air. Sebum goes through similar changes. The oily material released from sebaceous glands contains several kinds of lipids, which are fat-like molecules. Skin microbes can break apart or modify those lipids. Some of the resulting chemicals evaporate more easily than the original material and become part of the scent surrounding the body. Corynebacterium species are especially common in moist areas such as the armpits, where they can help produce familiar body odors from compounds in skin secretions. Cutibacterium species are well adapted to oily areas and can use lipids found in sebum. These microbial processes help explain why human scent differs so much across the body. The chemical environment around an ankle can differ from the environment around an arm. Feet are particularly interesting because they contain many sweat glands and spend long periods in warm, enclosed footwear. They also support distinctive microbial communities. Some mosquito species, including certain malaria mosquitoes in the genus Anopheles, often approach lower parts of the body. Researchers have investigated whether odors associated with feet and ankles help guide that behavior. Experiments with Anopheles mosquitoes have also found relationships between skin bacteria and how attractive different people are. In one line of research, people with different skin bacterial communities produced different levels of attraction when their odors were presented to mosquitoes. Certain bacterial patterns were associated with stronger attraction. That does not point to one special bacterium that makes a person irresistible. A mosquito encounters a mixture created by many organisms, glands, and chemical reactions working at the same time. The skin microbiome itself is also shaped by many influences. Bathing can temporarily change the chemicals present on the surface. Cosmetics and soaps can add new compounds. Climate affects temperature and moisture. Genetics and immune function can influence the kind of environment the skin provides for microbes. Diet and general physiology may also alter some secretions reaching the surface. These effects help explain why a person's scent can shift over hours or days, while still keeping some longer-lasting features. A shower may remove sweat, oils, and some surface microbes for a time. It does not reliably transform someone who consistently attracts mosquitoes into someone mosquitoes consistently ignore. The underlying skin continues producing secretions, and microbial communities begin rebuilding their usual chemical environment. Human odor therefore emerges from a partnership between the body's own chemistry and the microscopic organisms occupying its surface. For researchers, that creates a difficult problem. To prove that mosquitoes truly prefer one person's scent over another, they needed a way to separate odor from body heat, breath, movement, and appearance. Eventually, they found a simple material that could collect those invisible chemicals and carry them into the laboratory. Researchers eventually found a practical way to separate human scent from the person producing it. At Rockefeller University in New York City, Maria Elena de Obaldia worked with neuroscientist Leslie Vosshall and other researchers to measure how strongly mosquitoes preferred the odor of different people. Their results were published in the journal Cell in 2022. The team focused on Aedes aegypti, a mosquito species that has become especially well adapted to feeding on humans. Instead of placing volunteers directly inside cages with mosquitoes, the researchers collected the chemicals leaving their skin. Participants wore nylon sleeves over their forearms. Nylon can absorb some of the compounds released from the skin, allowing a piece of fabric to carry a person's scent after it has been removed. Those scented samples could then be presented to mosquitoes under controlled laboratory conditions. Female Aedes aegypti were given choices between nylon samples from different participants. This removed many of the cues that normally help mosquitoes locate people. The samples did not breathe out carbon dioxide. They had no warm skin, movement, or visible body shape. What remained was the chemical material collected from the person's skin. The mosquitoes showed clear preferences. Some samples attracted many more females than others. More importantly, the ranking did not disappear after a single test. Researchers repeated comparisons over long periods, and people who were strong attractors tended to remain strong attractors. Some participants kept roughly the same relative position through testing that extended across several years. That persistence offered laboratory support for something many people had noticed in ordinary life. Certain people really can produce a skin odor that Aedes aegypti finds consistently more attractive. The team then examined the chemistry collected from those participants. Highly attractive people tended to have greater amounts of several carboxylic acids on their skin. Carboxylic acids are a large family of organic compounds defined by a particular chemical group in their structure. Many occur naturally on human skin. Some longer-chain carboxylic acids are connected with the oily material produced by sebaceous glands and with chemical reactions taking place at the skin surface. The pattern was strong enough to make these acids an important part of the mosquito magnet explanation. The researchers also tested mosquitoes whose odor-sensing systems had been altered genetically. Part of mosquito smell depends on proteins called ionotropic receptors. These proteins help sensory nerve cells respond to particular chemicals in the air. Three important helper proteins in this system are called IR8A, IR25A, and IR76B. Researchers produced mosquitoes missing one of these helper proteins and compared their behavior with ordinary Aedes aegypti. Mosquitoes with these sensory changes showed a major reduction in their overall attraction to human odor. Their chemical world had become harder to read. Even so, some of these altered mosquitoes could still distinguish more attractive human samples from less attractive ones. That result showed how much backup exists within the mosquito's sensory system. Human scent contains many chemicals, and mosquitoes possess several ways of detecting them. Carboxylic acids appear to contribute strongly to persistent attraction, while the complete attractive mixture remains more complicated. There was no single molecule in the experiment that could be labeled the universal mosquito magnet chemical. One participant could have more of several acids and attract many mosquitoes, while another person's overall chemical mixture produced a weaker response. The important discovery was the stability of the difference. Soap, meals, weather, and daily activity can alter what reaches the skin for a time. Yet some people continued producing an odor profile that mosquitoes preferred year after year. A trait that persistent points toward deeper influences within human biology, including characteristics that may be partly inherited. Long-lasting differences in mosquito attraction suggest that some of the cause may be built into a person's biology. One way scientists can study that possibility is by comparing twins. Identical twins begin from a single fertilized egg that later splits into two embryos. Because of that shared origin, they inherit nearly the same DNA sequence. Fraternal twins develop from two separate eggs, fertilized by two separate sperm cells. Genetically, they are much more like ordinary brothers or sisters, sharing about half of the inherited genetic differences that vary among people. That contrast gives researchers a useful natural experiment. If identical twins resemble each other more strongly in a particular trait than fraternal twins do, inherited biology may be helping shape that trait. Researchers applied this method to mosquito attraction in a study involving 18 pairs of identical female twins and 19 pairs of fraternal female twins. The volunteers were tested using Aedes aegypti mosquitoes. Air carrying human scent was directed through a branching device that allowed the mosquitoes to move toward one odor source or another. Scientists then measured how attractive each participant was. Mosquito attractiveness was more similar within identical twin pairs than within fraternal twin pairs. That pattern supported the idea that genetics contributes to how strongly mosquitoes respond to a person. Scientists often describe this kind of influence using the word heritability. Heritability refers to how much of the variation in a trait across a particular population can be linked to inherited genetic differences. It does not mean that a person's result is permanently fixed by DNA. Height offers a familiar example. Genes strongly influence height, yet childhood nutrition and health can also affect how tall someone becomes. Mosquito attraction appears to work through a similar mixture of inherited biology and environmental influence. The mosquito itself never detects a person's genes. DNA remains inside human cells and provides instructions used to build and regulate the body. Those instructions can influence traits that eventually change the signals reaching a mosquito. Genes can affect how glands in the skin work. They may influence metabolism, immune activity, the amount and type of substances released onto the skin, and the conditions in which different skin microbes grow. Each of those effects can eventually alter the volatile chemicals leaving the body. Volatile simply means that a substance can enter the air easily enough to be detected as part of a scent. Larger genetic studies have also searched the human genome for places associated with mosquito attraction. A genome-wide association study compares genetic variation across many people and looks for small DNA differences that appear more often among people sharing a trait. One study involving thousands of participants identified several candidate regions associated with how attractive people believe themselves to be to mosquitoes. Those findings provide clues rather than a complete map. Scientists still cannot trace most of those genetic differences through a simple chain from one gene to one skin chemical, and then to a mosquito's antenna. The 2022 odor experiments also showed why that simple explanation would probably be incomplete. Highly attractive people tended to carry greater amounts of several carboxylic acids on their skin, while the mosquito was responding to a much larger chemical mixture. Inherited traits could help shape that mixture through several biological routes at once. Daily life continues changing it as well. Weather affects how much a person sweats. Bathing changes the skin surface for a time. Food, activity, hormones, cosmetics, and local microbes can all influence the chemicals present around the body. A person who has seemed especially attractive to mosquitoes for years may therefore have a genuine biological tendency underneath those changing daily conditions. That tendency still rises and falls with physiology. A quiet body sitting at rest sends a different combination of signals from the same body after exercise, during pregnancy, or after spending time in summer heat. A person's usual level of mosquito attraction can stay fairly stable for years, while the signals leaving the body still change from hour to hour. Breathing rate shifts with activity. Skin temperature rises and falls. Sweat changes the moisture and chemistry around exposed skin. These temporary changes are layered onto the longer-lasting odor profile shaped by skin chemistry and biology. Exercise shows this especially clearly because it changes several mosquito cues at the same time. Working muscles need more energy than resting muscles. As metabolism increases, cells consume more oxygen and produce more carbon dioxide. The bloodstream carries that extra carbon dioxide to the lungs, and breathing becomes faster or deeper to remove it. A person who has just finished a run can therefore release more carbon dioxide than the same person sitting quietly. Muscle activity also produces heat. Blood flow toward the skin can increase as the body tries to release some of that heat into the surrounding air. When cooling requires more help, sweat glands begin moving fluid onto the skin. As sweat evaporates, it removes heat. The remaining moisture also changes the small layer of air directly above the skin. Sweat contains salts and organic compounds that can later be altered by skin microbes. One substance that has received decades of attention in mosquito research is lactic acid. Lactic acid and closely related lactate chemistry increase during intense exercise and are also associated with normal human skin emissions. Experiments with Aedes aegypti have shown that lactic acid can strengthen attraction when it appears alongside other human signals. Its effect depends on the larger odor mixture, so it works as one ingredient within a more complicated chemical signal. Pregnancy can change mosquito attraction through several physiological routes as well. Pregnancy gradually raises the body's energy requirements as maternal tissues support the developing fetus. Ventilation also changes. By late pregnancy, a woman may breathe a greater volume of air each minute than before pregnancy, which can increase the amount of carbon dioxide released. Blood flow to the skin can rise, and body temperature can shift slightly. Studies carried out in malaria endemic regions have found that pregnant women sometimes attract more Anopheles mosquitoes than non-pregnant women. One well-known study in the Gambia found pregnant women attracting roughly twice as many Anopheles gambiae mosquitoes during experimental collections. That does not mean pregnancy guarantees more bites. Local mosquito species, clothing, sleeping conditions, repellents, weather, and individual odor chemistry continue to affect what happens. Alcohol has also appeared in mosquito studies, although the evidence here is much less settled. A small experiment published in the early 2000s tested volunteers before and after drinking beer. After beer consumption, more mosquitoes were attracted to some participants. Other small studies have reported similar patterns. Researchers have proposed possible changes in skin temperature, odor, or metabolism, yet no single mechanism has been firmly established. The size of these studies has also been modest, compared with the stronger evidence behind carbon dioxide sensing and persistent differences in skin odor. Everyday mosquito attraction is therefore flexible within limits. A person who naturally produces an odor that Aedes aegypti strongly prefers may remain a high attractor across many years. On a particular evening, exercise could make that person's breath easier to follow and skin warmer. Pregnancy can alter several physiological cues for months. Other temporary influences may have smaller effects that depend heavily on the mosquito species and setting. This mixture of stable traits and changing conditions helps explain why personal mosquito stories can become so convincing. Someone eats a particular food and gets bitten. Someone wears a certain shirt and notices more mosquitoes. Another person remembers that mosquitoes always seem to choose people with a certain blood type. Real changes are happening around the body all the time. which makes it easy to connect a memorable bite with whatever happened shortly before it. Testing those explanations under controlled conditions reveals that some familiar mosquito beliefs have much stronger support than others. The phrase "sweet blood" has been used for generations to explain why mosquitoes seem to prefer certain people. Scientifically, it has no clear meaning. Blood sugar, or glucose, circulates inside blood vessels beneath the skin. A mosquito flying several meters away has no way to measure that glucose before choosing a person. By the time its mouthparts reach blood, the insect has already followed breath, body-associated warmth, visual information, and skin odor to reach that particular host. Blood type has a little more scientific evidence behind it. Human blood is commonly classified using the ABO system. Someone may have type A, type B, type AB, or type O blood, depending partly on molecules called antigens found on the surface of red blood cells. Some mosquito experiments have reported different levels of attraction among these groups. A frequently discussed study using Aedes albopictus, the Asian tiger mosquito, found greater landing activity on people with type O blood than on some other blood groups. Researchers have also examined a trait called secretor status. Many people release substances related to their ABO blood group antigens into fluids such as saliva and other body secretions. These people are called secretors. Others release much less of this material. That difference offers a possible route by which blood group biology could influence chemicals available at the body surface before a mosquito feeds. The evidence remains limited. Studies have often involved relatively small groups of people, particular mosquito species, and controlled laboratory conditions. The much larger differences seen in direct human odor experiments point strongly towards skin chemistry as a major part of persistent mosquito attraction. Blood type may contribute in some circumstances, while it explains only a small part of the larger sensory picture. Food has produced its own collection of mosquito stories. Garlic is one of the oldest familiar examples. Its strong sulfur-containing compounds can affect human breath and body odor after eating it, which makes the idea sound reasonable. Controlled testing has not shown garlic consumption to provide dependable protection from mosquito bites. Vitamin B has also been promoted for decades as a way to change a person's scent. Experiments testing vitamin B supplements have failed to establish them as a reliable mosquito repellent. What a person wears can have a more direct effect because mosquitoes use vision while approaching a host. Aedes aegypti can respond strongly to objects that stand out against their surroundings. Experiments have shown that darker, high-contrast targets can receive more attention under some conditions. Clothing therefore changes how easy a body is to locate visually and how much skin remains exposed. It does not erase the underlying odor profile leaving that person's skin. Fragrance creates another complicated case. Perfume, scented lotion, soap, and shampoo add volatile chemicals to the cloud around the body. Some individual fragrance ingredients can attract or repel certain mosquitoes in laboratory tests. A finished perfume contains many compounds, however, and its effect can change as those compounds evaporate at different rates. Using fragrance alone has never become a dependable general method of preventing mosquito bites. Bathing also changes the skin surface temporarily. It can remove sweat, oils, environmental material, and some odor-producing compounds. Soon afterward, sweat glands resume secretion. sebaceous glands continue producing oil, and the skin's microbial community remains active. A person's characteristic chemistry gradually reappears. This is why mosquito attraction is better understood through levels of evidence. Carbon dioxide sensing has been demonstrated directly. Heat and visual responses can be measured under controlled conditions. Stable differences in human odor have been reproduced across repeated experiments, with skin chemistry showing especially strong links to persistent attraction. Blood type and several everyday habits may modify the result in particular settings, though their effects are smaller or less certain. Conflicting mosquito advice also has another source. Aedes aegypti is only one mosquito among thousands, and a species adapted to humans can respond very differently from one that normally searches for birds, deer, or other animals. The mosquito circling an ankle represents only one small branch of a remarkably diverse insect family. More than 3,500 mosquito species have been described around the world. They occupy habitats ranging from tropical forests to Arctic wetlands, and many rarely interact with people at all. Only a small fraction regularly creates serious problems for humans. Even among the species that do bite people, host preferences can differ greatly. Aedes aegypti is one of the clearest examples of a mosquito closely associated with humans. It commonly breeds around houses and cities, using artificial containers such as buckets, water tanks, flower pots, and discarded tires. In many parts of its range, adult females strongly prefer human blood. Scientists describe that preference with the word anthropophilic, which simply means attracted to humans as hosts. Aedes aegypti is also strongly active during daylight hours. Biting often increases in the morning and again later in the afternoon, although the exact pattern changes with temperature and local conditions. A closely related mosquito, Aedes albopictus, behaves somewhat differently. It is commonly known as the Asian tiger mosquito because of the pale stripes crossing its dark body. This species originated in Asia, but has spread widely through international trade, especially through shipments involving used tires and other objects that can hold water. Aedes albopictus readily bites humans, while its feeding range also includes other mammals and birds. That broader diet makes it less tightly specialized on people than Aedes aegypti. A different group appears in the genus Anopheles. Several Anopheles species are responsible for transmitting the Plasmodium parasites that cause human malaria. There are hundreds of known Anopheles species, although only a smaller number are important malaria vectors. Many of the most important human-biting species are active from evening through the night. Anopheles gambiae in sub-Saharan Africa is especially well known for its strong preference for humans. Other Anopheles mosquitoes feed more often on cattle or other animals. Scientists use the term zoophilic for species or populations that prefer non-human animals. Culex mosquitoes add another feeding pattern. Many Culex species frequently take blood from birds. That behavior is important in the natural cycle of West Nile virus, which can move between infected birds and mosquitoes. Some Culex mosquitoes also feed on mammals, including humans. Their host choices can even shift with season or location as the available animals change. Time of day varies just as much as host choice. Aedes aegypti commonly searches for hosts during daylight. Many Culex species become especially active around dusk and during the night. Important Anopheles malaria vectors often feed after sunset. These differences influence where people encounter mosquitoes and which protection methods work best. Resting behavior varies as well. Some species commonly enter buildings after feeding and rest on indoor walls. Others remain outside among vegetation or sheltered natural surfaces. A mosquito that frequently feeds indoors faces a different environment from one searching for hosts in woodland or marsh habitat. Those differences accumulate over generations. Natural selection favors sensory systems that work well with the animals and habitats available to each mosquito population. A species that depends heavily on humans benefits from recognizing human skin odor with great accuracy. A mosquito feeding mainly on birds gains more from sensory signals associated with birds. Generalists must remain flexible enough to respond to several possible hosts. This helps explain why mosquito advice gathered from different places can sound contradictory. The mosquito visiting a backyard in Florida may belong to a different species from one encountered beside a Canadian wetland or inside a home in East Africa. Its preferred host, active hours, and response to human odor may all differ. Across this diversity, the mosquitoes arriving at vertebrate skin for a blood meal share an important biological feature. They are females. The reason females seek blood reaches beyond ordinary feeding and into the production of eggs. For most of an adult mosquito's daily energy needs, plant sugar does the work. Male mosquitoes feed on nectar and other sugary plant fluids. They do not take blood meals from people, birds, or other vertebrate animals. Female mosquitoes also visit flowers and use sugar as fuel. Sugar provides energy for flight, movement, and basic metabolism. Blood serves a different biological purpose for females of many species. It supplies concentrated nutrients used to produce eggs. A blood meal contains large amounts of protein. During digestion, those proteins are broken into smaller building blocks called amino acids. The mosquito absorbs these amino acids through its digestive system and uses them to make new proteins inside the developing eggs. Blood also provides iron and other nutrients involved in egg development. For Aedes aegypti, this link between blood and reproduction is especially strong. A female usually begins seeking a host only after mating and reaching the proper reproductive state. Once she feeds successfully, her behavior changes. The abdomen fills and expands as blood enters the digestive system. Host-seeking activity then drops for a time, while digestion and egg development take priority. Inside the mosquito, chemical signals begin coordinating that change. Hormones are molecules that carry instructions between tissues. After feeding, hormonal signals help activate the ovaries and the mosquito's fat body. The fat body performs some jobs similar to those handled by the liver and body fat in mammals. It stores nutrients and produces important proteins. One of those products is vitellogenin, a large protein used to build egg yolk. After a blood meal, amino acids entering the mosquito help trigger vitellogenin production. The protein moves through the mosquito's internal fluid and is taken up by developing eggs inside the ovaries. Over the following days, those eggs grow. Temperature, species, and the size of the blood meal affect how quickly this happens. Under warm conditions, an Aedes aegypti female can often become ready to lay eggs within roughly 2 to 4 days after feeding. She then searches for a suitable place where her offspring can develop. Once the eggs have been deposited, another reproductive cycle may begin. Scientists call this repeating sequence the gonotrophic cycle, A female seeks blood, digests the meal while her eggs mature, lays those eggs, and later becomes ready to seek another host. Many females can complete several such cycles during their adult lives if they survive long enough. That repeated feeding matters especially for species such as Aedes aegypti. A female may encounter several different people across successive reproductive cycles. She can also interrupt a blood meal if she is disturbed and later continue feeding on another person. This habit helps explain why Aedes aegypti can be so effective at moving certain viruses between human hosts. There are exceptions within the mosquito family. Some females can produce an initial batch of eggs without taking a blood meal. That ability is called autogeny. It occurs in certain species or populations when enough nutrients have been stored during the larval stage. Later, egg batches may still require blood. This variation is another reminder that thousands of mosquito species do not all follow exactly the same reproductive pattern. For the familiar human-biting species, however, the connection between blood and egg production explains much of the behavior we experience. A female mosquito following carbon dioxide and human skin odor is carrying out one stage of a reproductive cycle. Reaching the host is only part of that task. The nutrients she needs are moving through vessels beneath a tough outer layer of skin, and reaching them requires one of the most specialized feeding structures found among insects. Once a female mosquito has landed, the feeding process becomes a matter of very small movements. From ordinary viewing distance, the proboscis projecting from her head looks like a single narrow needle. Under magnification, it turns out to be a bundle of specialized mouthparts working together. The part visible from the outside is called the labium. It forms a flexible sheath around the structures that actually enter the skin. During feeding, the labium bends backward and remains mostly outside the body. Inside that sheath is a bundle called the fascicle. A female mosquito's fascicle contains 6 slender stylets. A stylet is simply a narrow mouthpart shaped for piercing or handling tissue. 2 of the 6 are mandibles. Mosquito mandibles are extremely thin and help the feeding structures pass through the outer layers of skin. Another pair are called maxillae. The maxillae carry tiny serrated edges. These tooth-like surfaces help the mouthparts work their way forward with small movements rather than requiring one hard push. The remaining 2 stylets have especially important jobs once the mosquito is beneath the surface. One is the labrum. The labrum forms the main channel through which blood travels into the mosquito. The other is the hypopharynx. This narrow structure carries saliva in the opposite direction, from the mosquito into the feeding site. The entire arrangement is only a fraction of a millimeter across. Its movements are also more flexible than the appearance of the proboscis suggests. The stylets can bend slightly as they move through soft tissue. They can change direction beneath the surface while the mosquito searches for a suitable blood vessel. Small vessels called capillaries form dense networks throughout the skin. Larger vessels lie deeper. The mosquito probes through this tissue until its feeding structures reach blood that can be drawn efficiently. Sensory information helps guide that search. The tips of the mouthparts contain receptors that can respond to chemical and physical conditions inside the tissue. Once blood begins entering the labrum, pumps inside the mosquito's head provide the force needed to pull the liquid upward. These pumps are muscular structures connected to the feeding canal. Their repeated contractions create suction and move blood through the proboscis into the digestive tract. Saliva is being delivered during this process as well. That saliva contains a mixture of proteins and other molecules that help keep blood flowing long enough for the mosquito to feed. A vertebrate body normally responds to a damaged blood vessel through a system called hemostasis. Hemostasis is the set of processes that slows and stops bleeding. Blood vessels can narrow, platelets gather at the damaged area, clotting proteins help form a stronger seal. Mosquito saliva contains compounds that interfere with parts of this response. Some reduce platelet activity. Others affect clotting or the tightening of nearby blood vessels. Together, these effects give the female a brief period in which blood remains easier to draw through such a tiny feeding channel. A successful meal can be completed within minutes, although the exact time varies with mosquito species and whether the insect is disturbed. During that short interval, several structures have performed separate jobs inside an apparatus small enough to disappear against the skin. The labium supported the bundle from outside. The stylets created and explored the path through the tissue. The feeding channel then carried blood upward, while saliva entered through its own route. For the mosquito, those salivary compounds make the blood meal easier to obtain. For the human host, the same saliva begins an immune response that can remain noticeable long after the mosquito has flown away. Once mosquito saliva enters the skin, the human immune system begins responding to it. The saliva contains dozens of biologically active substances that help the female complete her blood meal. Some interfere with platelets, the tiny cell fragments that help form clots. Other salivary proteins affect blood vessels or parts of the clotting system. Together, these compounds temporarily make it easier for blood to keep flowing through the mosquito's narrow feeding channel. Human immune cells also encounter those unfamiliar salivary molecules. The body recognizes some of them as foreign material and begins a local immune response around the feeding site. One important participant is the mast cell. Mast cells are immune cells found in connective tissues, including large numbers close to the skin and blood vessels. They contain small storage packets filled with chemical messengers. When the immune system reacts to mosquito saliva, mast cells can release one especially familiar messenger called histamine. Histamine changes the tiny blood vessels near the bite. The walls of those vessels become more permeable, meaning that fluid and immune-related substances can pass through them more easily. Some fluid moves from the bloodstream into the surrounding tissue. That local buildup helps produce the small raised swelling commonly called a mosquito bite. Doctors often call this kind of raised patch a wheal. Histamine also stimulates nerve endings in the skin. Those nerves send signals toward the spinal cord and brain, producing the familiar sensation of itching. The mechanical puncture itself is therefore only one small part of what a person feels. Much of the irritation develops because the immune system has reacted to material left behind in the saliva. The timing can vary. A person may notice irritation while the mosquito is still nearby, or the bump may become much more obvious several minutes later. For some people, redness and swelling continue increasing for hours. The size of that reaction differs greatly from one person to another. Someone can develop a broad, noticeable swelling from a single feeding event. Another person may have a tiny bump that disappears quickly. Immune history helps explain some of that variation. Repeated mosquito exposure can change how strongly the body responds to salivary proteins. Young children encountering mosquitoes for the first times in their lives may react differently from adults who have experienced many bites from the same local species. Travel can also expose someone to mosquito species whose saliva contains a somewhat different mixture of proteins. The immune response may therefore look different from the response that person normally sees at home. This creates an important complication when people compare how badly mosquitoes seem to like them. 10 large, itchy bumps do not necessarily mean that one person received 10 times more mosquito attention than someone with smaller marks. One person's immune system may simply produce stronger visible reactions. Some people respond so mildly that individual bites are difficult to notice at all. Scientists studying mosquito attraction therefore prefer measurements such as approaches, landings, feeding attempts, or choices between controlled odor samples rather than counting itchy bumps afterward. Those methods separate the mosquito's decision from the human body's reaction to that decision. By the time an itchy wheal appears, the female may already be far from the person she fed on. Inside her abdomen, the blood meal is beginning to change. Water and salts must be managed, proteins must be digested, and nutrients from the meal will soon be directed toward the ovaries. The same feeding event that leaves a temporary patch on human skin has started a much larger reproductive process inside the mosquito. After feeding, the female mosquito carries a meal that is enormous compared with her own size. In Aedes aegypti, a full blood meal can approach or even exceed the mosquito's unfed body mass. Her abdomen stretches as the digestive tract fills, changing from a narrow shape into a much wider one within minutes. That sudden increase creates an immediate problem. Blood contains a large amount of water, along with salts and nutrients that the mosquito needs to process. Carrying all of that extra liquid makes flight more difficult and adds weight that provides little value for developing eggs. The mosquito begins correcting this surprisingly quickly. During feeding and shortly afterward, her kidneys, called Malpighian tubules, remove excess water and dissolved ions from the body fluid. This rapid fluid loss is known as postprandial diuresis. Postprandial simply means after a meal, while diuresis means producing and removing extra fluid. In some mosquito species, droplets of fluid can begin leaving the body before the blood meal has even been completed. Reducing that water load concentrates the nutrients that remain. Digestion then becomes the main task. The blood enters a digestive chamber called the midgut. Enzymes, called proteases, begin cutting large blood proteins into smaller pieces. Those pieces are eventually broken down into amino acids, the basic building blocks used to make new proteins. Hemoglobin, the oxygen-carrying protein inside red blood cells, makes up a large part of the material being processed. Digesting so much blood also releases iron-containing compounds that can become chemically harmful in high amounts. Mosquitoes have protective systems that bind, store, or isolate some of these materials while digestion continues. Over roughly the next 2 or 3 days under warm conditions, nutrients from the meal move out of the midgut and into other tissues. The ovaries begin changing at the same time. Each ovary contains developing egg cells called oocytes. Before feeding, these cells are small and only partly developed. A successful blood meal activates hormonal signals that tell the mosquito that enough nutrients are available to continue reproduction. One important hormone in Aedes aegypti is called 20-hydroxyecdisone. After a blood meal, signals connected with the ovaries help raise this hormone. It then helps activate genes in the fat body, an insect organ involved in storing nutrients and producing important proteins. Among the proteins produced, there is vitellogenin. Vitellogenin travels through the mosquito's internal fluid to the ovaries, where developing oocytes take it up. It becomes a major source of yolk material for the embryos that may later develop inside the eggs. As this process continues, the ovaries enlarge. The abdomen that first expanded because of blood gradually becomes occupied by developing eggs as digestion nears completion. Aedes aegypti females commonly produce dozens of eggs from one successful blood meal, although the number varies with body size, nutrition, temperature, and how much blood was obtained. Behavior changes along with the internal chemistry. A female that has fed fully usually becomes much less interested in human odor for a time. The sensory system that recently guided her toward carbon dioxide and warm skin is temporarily working within a different reproductive state. Resting becomes common while digestion proceeds. As the eggs approach maturity, her priorities shift again. The mosquito begins responding more strongly to cues associated with places suitable for laying eggs. Humidity, water, container surfaces, and chemical signals from potential breeding sites become increasingly important. Scientists call the act of laying eggs oviposition. For a female Aedes aegypti, finding an oviposition site can mean searching around a house or yard for an amount of standing water that a person might barely notice. A blood meal taken from a human arm has now been converted into the raw material for a new batch of mosquitoes. The search that began with breath and skin odor has turned away from the human body and toward water. Once her eggs are developing, the female mosquito begins paying attention to a very different set of signals. Warm skin and exhaled carbon dioxide become less important for a time. Her sensory system shifts toward finding a place where the eggs can survive after they are laid. Scientists call this behavior oviposition. Oviposition simply means laying eggs, and mosquito species have evolved several different ways of doing it. The phrase standing water is useful, although it can make mosquito breeding sites sound larger than they really are. For Aedes aegypti, a pond is often unnecessary. A small container holding rainwater can provide enough space for an entire generation to begin. Aedes aegypti females usually lay their eggs one at a time on damp surfaces just above the waterline. A bucket, flower pot, barrel, or discarded tire can all provide those surfaces. The eggs are tiny, dark, and only about 0.5 millimeter long. They often remain attached to the inside wall of the container while the water level drops. This gives Aedes eggs an important survival advantage. Many can withstand drying for months. When rain later raises the water level and covers the eggs, chemical and physical changes around them can trigger hatching. That ability helps explain why a container that has been dry for weeks can still produce mosquito larvae after it fills again. Culex mosquitoes use a different strategy. Many Culex females place their eggs directly on the water surface in groups joined together into floating rafts. A single raft may contain well over 100 eggs, depending on the species and the condition of the female. The eggs stand upright together, creating a small floating structure that can be seen with careful inspection. Anopheles mosquitoes generally lay their eggs individually on the water. Their eggs have specialized side structures that help them remain at the surface. These differences mean that mosquito eggs can occupy very different positions, even when the larvae that emerge from them all depend on water. The female also evaluates the quality of the site. Mosquitoes can respond to moisture, odors from microbes, decomposing plant material, and chemicals associated with water that has already supported other mosquitoes. Some species prefer relatively clean container water. Others do well in water rich in decaying leaves or organic waste. Shade can matter because exposed containers may heat up or dry out quickly. Vegetation can provide shelter. Salinity also matters, since some mosquito larvae tolerate salty or brackish water far better than others. Natural breeding sites can include tree holes that collect rain, temporary woodland pools, marsh edges, floodplains, or water trapped between the leaves of certain plants. Human environments add many new possibilities. Rain collects inside gutters that do not drain properly. Water remains in plant sources after watering. Discarded tires trap small pools beneath their curved inner walls. Buckets and barrels can hold water for weeks if they remain uncovered. Even small pieces of outdoor equipment can develop pockets where rain stays long enough for eggs to hatch. For a female, choosing among these sites directly affects the survival of her offspring. A container that dries too soon can kill aquatic larvae. Water with very little food can slow their growth. A site crowded with other larvae can increase competition. Predators may make some ponds dangerous even when they contain plenty of water. Oviposition, therefore, involves another form of sensory selection. The mosquito that recently distinguished one human odor from another now evaluates water, surfaces, chemicals, and local conditions before releasing her eggs. The scale can be surprisingly small. A few spoonfuls of water collected in a sheltered container may be enough for larvae to develop if the water remains there long enough. To a person, it can look like an insignificant puddle. For the young mosquito that hatches into it, that small volume of water becomes the entire environment in which its body begins to change. When a mosquito egg hatches, the animal that emerges looks very different from the winged insect it will eventually become. The young mosquito is an aquatic larva. It has no wings, does not search for blood, and spends this stage entirely in water. Mosquitoes go through 4 major life stages: egg, larva, pupa, and adult. This pattern is called complete metamorphosis. It means the juvenile body is extensively rebuilt before the adult form appears. A mosquito larva has a clearly visible head, a narrow thorax behind it, and a long segmented abdomen. Its movements are quick and flexible, which is why mosquito larvae are sometimes called wrigglers. Most spend much of their time feeding. Tiny brushes near the mouth can sweep microorganisms and suspended particles from the water. Depending on the species and habitat, the diet may include bacteria, algae, microscopic organisms, and fragments of decaying organic material. This makes a rain-filled container more biologically active than it may appear. Water containing fallen leaves can quickly develop communities of bacteria and other microorganisms that become larval food. As the larva grows, its outer covering cannot simply stretch forever. Mosquitoes have an exoskeleton, which means their firm supporting layer is on the outside of the body. Growth therefore requires shedding that old covering. The larval period is divided into 4 stages called instars. A mosquito begins as a first instar larva. After growing, it molts into the second instar, later reaches the third, and eventually becomes a fourth instar larva. Each molt provides space for a larger body. Breathing also shows how differently mosquito species use the water surface. Many Aedes and Culex larvae possess a breathing tube called a siphon near the end of the abdomen. They rise toward the surface and place the siphon into the air, often hanging downward beneath it. Anopheles larvae use a different arrangement. They lack the long respiratory siphon common in Aedes and Culex. Instead, they usually rest with much of the body lying roughly parallel to the water surface while taking in atmospheric oxygen. After the fourth larval stage, feeding stops and the mosquito becomes a pupa. The pupa has a curved, compact shape and is sometimes called a tumbler because it can move rapidly through the water when disturbed. Mosquito pupae remain surprisingly active even though they do not eat. They still breathe at the surface and can swim away from disturbances. Inside the pupal covering, the body is undergoing its largest transformation. Larval tissues are reorganized while adult structures develop. Wings take shape. Long adult legs form. The antennae, compound eyes, reproductive organs, and specialized mouthparts reach their mature arrangement. When development is complete, the pupal casing splits at the surface. The adult mosquito slowly pushes itself out and rests on the empty shell, or directly at the water surface. Its body is soft at first. The legs must become firm enough to support its weight, and the wings need time to expand and harden before normal flight begins. The speed of this entire process depends strongly on temperature, food, crowding, and species. Under warm, favorable conditions, some mosquitoes can move from egg to flying adult in roughly 1 to 2 weeks. Cooler water can slow development considerably. For an Aedes aegypti growing in a small summer container, the change can therefore happen within a period short enough for one spell of warm weather to produce a new group of adults. The insect that leaves the water now enters a completely different environment. Its juvenile life depended on microscopic food and access to the water surface. As an adult, flight opens a wider world of flowers, sheltered resting places, potential mates, and, for females in the proper reproductive state, vertebrate hosts. Blood feeding will become important again, but much of adult mosquito life is spent doing something else entirely. For an adult mosquito, blood feeding occupies only part of a much broader life. Flight itself requires a steady supply of energy. That energy usually comes from sugar. Male mosquitoes depend entirely on plant sugars and other carbohydrate-rich liquids because they never take blood meals. Females also visit flowers and feed on nectar throughout adulthood, including between reproductive cycles. Nectar contains sugars such as sucrose, glucose, and fructose. After being absorbed through the digestive system, those sugars can be broken down to provide energy for flight, movement, and basic cell function. A mosquito may also obtain sugar from damaged fruit, plant secretions, or honeydew, the sugary liquid produced by insects such as aphids. Flower visiting sometimes gives mosquitoes a second ecological role. While reaching into a flower for nectar, pollen can attach to the mosquito's body and later reach another flower. One unusually well-studied example involves the blunt-leaved orchid Platanthera obtusata, which grows across northern parts of North America and Eurasia. Researchers working in northern Washington state have watched Aedes mosquitoes visit these orchids and carry packets of pollen attached near their eyes. Experiments published in 2020 showed that mosquito visits could successfully pollinate the flowers. Both male and female mosquitoes participated. The orchid produces a scent rich in a chemical called nonanol, which helps attract certain Aedes mosquitoes toward its flowers. For adults that survive long enough to reproduce, mating is another major part of life. In many mosquito species, males gather in loose aerial swarms. These swarms often form near visible markers in the landscape, such as the edge of a bush or another object that provides a reliable meeting place. Females enter the swarm and may pair with individual males. Sound helps at close range. The familiar mosquito buzz comes mainly from wings beating hundreds of times each second. Male mosquitoes generally have large feathery antennae that are especially sensitive to the flight tones produced by females. Research on Aedes aegypti has shown that males and females can adjust their wing beat frequencies during courtship. Their tones may approach shared harmonic frequencies, meaning that higher multiples of their separate wing beat sounds begin to line up. This acoustic interaction can help the insects recognize and track one another during flight. After feeding or mating, adults spend considerable time resting. Aedes aegypti often shelters in shaded, protected places around homes and vegetation. Other species rest beneath leaves, on walls, or in humid spaces protected from direct sun and wind. Resting helps conserve energy and reduces exposure during the hottest or driest parts of the day. Activity patterns differ greatly among species. Some Aedes mosquitoes are strongly active during daylight. Many Anopheles species become more active after sunset. Culex mosquitoes often show strong evening and nighttime activity. Survival in any of these periods is uncertain. Adult mosquitoes can be caught by spiders, dragonflies, birds, bats, and other insect-eating animals. Aquatic predators affect the earlier stages as well, including predatory insects and fish in habitats where fish are present. No single predator controls every mosquito population. Local habitat, weather, food availability, and species composition all influence how many adults survive. Lifespan varies just as widely. Under favorable laboratory conditions, some adult mosquitoes can survive for several weeks. Wild mosquitoes often face harsher conditions from dehydration, storms, predators, and limited food, shortening that span considerably. During whatever time remains, the adult shifts among feeding, resting, mating, and reproduction, according to its sex and reproductive state. That broader life existed long before mosquitoes became closely tied to human homes. For much of their evolutionary history, humans were simply one possible vertebrate among many. The unusually close relationship between people and species such as Aedes aegypti developed later, as some mosquito populations began finding both food and breeding opportunities around human settlements with remarkable reliability. The close relationship between Aedes aegypti and humans has deep roots in Africa. The species' ancestral populations are associated with sub-Saharan Africa, where many still live in natural habitats and feed on a wider range of animals. Some of these forest-associated mosquitoes readily choose non-human mammals when given the opportunity. Populations found around towns and cities can behave very differently. The strongly domesticated form of Aedes aegypti has become unusually good at living beside people. Human odor attracts these mosquitoes, and their larvae often develop in containers created or maintained by humans. That combination may help explain how the partnership began. As permanent settlements became more common over the past several thousand years, people began keeping water close to where they lived. Clay vessels, storage jars, wells, and later barrels or tanks could hold water through long dry periods. For a mosquito whose young require water, those containers offered something extremely valuable. Natural pools may disappear during a long dry season. Stored household water can remain available every day. A mosquito using those containers would also grow up surrounded by humans. Once the adults emerged, people provided a large and predictable supply of blood nearby. Natural selection could favor mosquitoes that were especially good at detecting the odor of those abundant hosts. A large study published in 2020 examined how strongly this pattern still varies across Africa. Researchers collected Aedes aegypti from 27 locations across sub-Saharan Africa. They established laboratory populations and tested thousands of female mosquitoes with human odor and animal odor. Some populations showed little interest in humans. Others strongly preferred human scent. Mosquitoes from parts of East Africa generally leaned more toward animal odors, while some populations in West Africa, especially Senegal, showed much stronger human preference. The researchers then compared those behaviors with the environments where the mosquitoes had originally been collected. Two factors explained most of the geographic pattern. One was human population density. Areas containing more people tended to produce mosquitoes with a stronger preference for human odor. The second factor was the intensity of the dry season. Places with long, hot periods of very little rainfall also tended to contain more human-preferring mosquitoes. Together, those ecological factors explained about 83% of the variation the researchers observed. Dry season intensity accounted for the larger share. That result fits closely with the stored water idea. In a region where rainwater disappears for months, a mosquito that depends on natural pools faces a difficult period. A population adapted to breeding inside human water containers can continue reproducing much closer to a reliable source of water. Humans then become a reliable source of blood in exactly the same place. The team also examined mosquito DNA. They sequenced the genomes of 375 individual mosquitoes collected across the study area. Human preference was associated with a particular pattern of genetic ancestry, and several chromosome regions showed especially strong differences between human-preferring and animal-preferring populations. Behavior and environment had therefore left measurable traces in mosquito genetics. This evolutionary change probably developed gradually, with important parts of human specialization emerging within roughly the past 10,000 years. That time range overlaps with the growth of larger permanent human settlements, although scientists cannot assign the beginning of human preference to one precise year or one village. The process also continues. Cities provide dense concentrations of people, along with countless small water containers. The 2020 study projected that continuing urban growth could favor stronger human preference in additional African Aedes aegypti populations by around 2050. A mosquito population living beside people therefore experiences a powerful combination of opportunity and selection. Reliable water helps its young survive. Dense settlements make humans easier to find than scattered wildlife. Generations of mosquitoes that respond efficiently to human odor can leave more descendants in that environment. For Aedes aegypti, this history transformed certain populations into some of the most human-focused mosquitoes on Earth. That specialization also changed what a blood meal could accomplish. A female moving repeatedly between human hosts can carry biological passengers with her, linking one person's bloodstream with another through the mosquito's own body. A mosquito that has taken blood from an infected host does not automatically become dangerous to the next animal it visits. Most individual mosquitoes are not carrying a human pathogen. Even when a mosquito feeds on infected blood, successful transmission requires a very specific biological match. Scientists use the word vector for an organism that can carry a pathogen from one host to another. A mosquito becomes an effective vector only when the pathogen can survive inside that particular mosquito species and complete the steps needed for later transmission. This biological ability is called vector competence. A mosquito may ingest a virus or parasite and still fail to transmit it because the microorganism cannot escape the gut, reproduce effectively, reach the salivary glands, or survive long enough inside the insect. Malaria provides one of the clearest examples of how complicated this relationship can be. Human malaria is caused by single-celled parasites in the genus Plasmodium. These parasites depend on mosquitoes in the genus Anopheles for an essential part of their life cycle. When a suitable female Anopheles feeds on a person carrying transmissible stages of Plasmodium, parasites enter the mosquito's midgut with the blood. They undergo several developmental changes inside the mosquito. Some eventually cross the gut wall and form structures on its outer surface. After further development, large numbers of slender forms called sporozoites are produced. Sporozoites are the stage capable of beginning a new human infection. They travel through the mosquito's body and eventually enter the salivary glands. Only after this sequence has been completed can a later blood-feeding event provide a route into another person. Mosquito-borne viruses follow a different biological path. Dengue, Zika, yellow fever, and chikungunya are strongly associated with Aedes mosquitoes, especially Aedes aegypti. After an Aedes female takes an infectious blood meal, virus particles first encounter cells lining the midgut. A successful virus must infect those cells and reproduce. New virus particles then spread through mosquito tissues. Eventually, some reach the salivary glands, where further replication can make transmission possible during a later feeding event. This process takes time. The interval between a mosquito acquiring a pathogen and becoming able to transmit it is called the extrinsic incubation period. For many mosquito-borne viruses, this can take roughly a week or longer, depending heavily on temperature, mosquito species, and the particular virus. Warmer conditions within a suitable range can speed viral replication. Temperature also affects how quickly mosquitoes develop, how often they feed, and how long adults survive. A mosquito must remain alive through enough of the incubation period for transmission to become possible. Culex mosquitoes illustrate a different transmission system. Several Culex species are important vectors of West Nile virus. Birds are central to the natural cycle. A mosquito feeds on an infected bird, becomes infected if it is biologically competent, and may later transmit the virus to another bird. Humans and horses can also become infected when certain Culex mosquitoes feed on them. In most circumstances, humans and horses develop too little virus in their blood to infect many new mosquitoes. For that reason, they are often described as incidental or dead-end hosts in the West Nile transmission cycle. These examples explain why identifying the mosquito species matters so much. Anopheles biology is central to malaria. Aedes aegypti has an especially important relationship with several human viruses. Culex species frequently connect mosquito populations with birds and occasionally other vertebrates. Each system depends on a chain of compatible organisms. An infectious host must first encounter a suitable mosquito. The pathogen must then survive and develop or reproduce inside that insect. Later, the mosquito must find another susceptible host, while it is still capable of transmitting the pathogen. A mosquito species that strongly prefers humans creates more opportunities for that final link whenever the pathogen also depends heavily on human hosts. The same sensory specialization that helps Aedes aegypti locate human odor can therefore influence how often human to mosquito to human contact occurs. And because mosquitoes already divide their attention unevenly among individual people, those opportunities may also be distributed unevenly within a household or community. The unequal attention that seems obvious when two people sit beside each other can remain important when the scale expands to an entire community. Field studies have found that mosquito contact is often distributed unevenly among people. Some individuals receive far more bites than others living in the same general environment. Researchers sometimes describe this pattern as heterogeneous biting. Heterogeneous simply means unevenly distributed. In some settings, a relatively small part of the population can account for a disproportionately large share of mosquito feeding. That matters because many early mathematical models of mosquito-borne disease made a simplifying assumption. They treated people as though each person had roughly the same chance of being bitten. Real communities contain much more variation. One household may have open windows and no screens. Another may use insecticide-treated bed nets every night. People differ in the amount of time they spend outdoors, the clothing they wear, and the places where they sleep. Mosquitoes add their own preferences on top of those differences. A person producing a highly attractive odor profile can become connected to mosquitoes more frequently than a nearby person whose scent produces a weaker response. Mathematical models can include this by giving individuals different contact rates. When some people are bitten repeatedly, transmission pathways become concentrated around those frequent mosquito-human encounters. Attractiveness remains only one part of the calculation. Mosquito abundance strongly affects how many encounters can occur. The fraction of mosquitoes carrying a pathogen also matters. Human immunity can change the chance that an exposure produces infection. Housing, access to bed nets, local climate, healthcare, and the biological competence of the mosquito species all influence the eventual outcome. Someone who attracts many mosquitoes, therefore, does not automatically become what is sometimes called a super spreader. They would also need to be infectious at the relevant time and capable of passing enough pathogen back to competent mosquitoes. Attractiveness, infectiousness, and susceptibility describe separate parts of the transmission network. Malaria research has revealed an especially interesting connection between these parts. Studies involving Plasmodium infection have found that mosquitoes can sometimes become more attracted to infected people during particular stages of infection. Researchers have investigated the volatile chemicals leaving the skin and breath of those hosts. Some studies have reported increased amounts of aldehydes. Aldehydes are a family of volatile organic compounds, several of which have noticeable odors even at low concentrations. Certain malaria-associated odor mixtures have produced stronger mosquito responses in laboratory or field experiments. The effect has not appeared identically in every study or every infected person. Age, parasite stage, mosquito species, and local conditions can influence the result. Scientists therefore treat the idea of parasite-driven attraction carefully. The evidence supports a biological possibility in which infection changes human chemistry in ways that can increase mosquito attraction under certain circumstances. There is no need for the parasite to make any conscious decision. Natural selection can preserve parasite traits that happen to improve movement between the organisms required for reproduction. For Plasmodium, that movement depends on both humans and Anopheles mosquitoes. A change in host odor that increases mosquito contact can therefore alter the parasite's opportunities to continue its life cycle. This returns the mosquito magnet question to the same skin chemistry that began the story. A difference in carboxylic acids or other odor compounds may determine which person receives more attention on a summer evening. Across thousands of people, repeated differences in contact can help shape the structure through which mosquito-borne pathogens move. Public health researchers therefore care about mosquito attraction for reasons extending beyond reducing itchy bites. Understanding who is bitten most often, which chemicals attract mosquitoes, and how those signals change could improve models of transmission and help identify better ways to interrupt contact. Once the mosquito search is broken into individual steps, control becomes much more precise. Scientists can interfere with host detection, reproduction, or the mosquito population itself, rather than treating every part of its life as a single problem. Once scientists separate mosquito behavior into individual biological steps, each step becomes a possible place to interfere. At the skin, repellents can make successful host finding and feeding less likely. DEET has been used as an insect repellent since the 20th century, while picaridin became another widely used option. These chemicals do not make a person's carbon dioxide, warmth, and skin odor disappear. They alter the mosquito's sensory interaction with treated skin, reducing the chance that an approach ends in feeding. Physical barriers provide another layer. In regions where malaria mosquitoes feed mainly at night, insecticide, treated bed nets place fine mesh between a sleeping person and the mosquito. The insecticide adds chemical protection to that physical barrier and has become one of the major tools used in malaria control. Other interventions target the mosquito before adulthood. Removing water from buckets, tires, uncovered containers, and other breeding sites can prevent Aedes aegypti larvae from completing development. This approach is called source reduction. Larval control can also use biological or chemical treatments in water that cannot simply be emptied. Public health programs need to know where mosquitoes are before choosing any of these methods. Adult traps can measure flying populations. Ovitraps provide small egg-laying sites that reveal the presence of container-breeding mosquitoes. Larval surveys examine standing water directly. Collected mosquitoes can then be identified by species, while some surveillance programs test them for particular pathogens. Modern control has also begun exploiting mosquito reproduction itself. Wolbachia is a group of bacteria naturally found inside many insect species. Aedes aegypti does not normally carry the Wolbachia strains used in current control programs, so scientists can introduce them deliberately. One strategy releases both male and female mosquitoes carrying particular Wolbachia strains. Females pass the bacterium to their offspring through their eggs. Over generations, Wolbachia can spread through the local mosquito population. Certain strains also reduce the ability of Aedes aegypti to transmit viruses such as dengue. This approach is called population replacement because the aim is to establish mosquitoes carrying Wolbachia rather than simply eliminate the local population. A separate strategy uses Wolbachia to suppress mosquito numbers. Here, programs repeatedly release Wolbachia-infected males. When those males mate with ordinary wild females, a reproductive effect called cytoplasmic incompatibility can prevent the embryos from developing successfully. The released males do not bite because male mosquitoes do not take blood meals. Repeated releases can gradually reduce the number of local Aedes aegypti. Singapore has tested this suppression method on an unusually large scale. A cluster randomized trial published in 2026 divided 15 geographic areas into 8 intervention clusters and 7 control clusters. More than 724,000 residents lived across those areas. After 3 months of releases, the average abundance of wild female Aedes aegypti remained far lower in the intervention areas throughout the 24-month trial. Depending on how long residents had been exposed to the program, researchers calculated roughly 71 to 72% protection against symptomatic dengue. Other population control methods use sterile males produced through irradiation or genetic techniques so that matings produce few or no viable offspring. Each method targets a different weakness in mosquito biology. Sensory research is creating additional possibilities. The 2022 experiments linking persistent human attraction with carboxylic acids revealed chemical pathways that future repellents or mosquito attractants might exploit. Researchers have not yet turned that discovery into a proven consumer product that simply blocks a person's mosquito magnet chemistry. The 2024 discovery that Aedes aegypti can use thermal infrared at close range offers another possible tool. Future traps may become more convincing by combining odor, carbon dioxide, visible contrast, and body-like thermal signals in carefully controlled ways. Mosquito control therefore becomes a collection of precisely chosen interruptions. A barrier can block feeding. A removed container can stop development, and reproductive biology can reduce future generations. Understanding how the mosquito finds us reveals where that search can fail. After following those mechanisms from human breath to mosquito reproduction, the familiar choice between two nearby people can finally be seen as the product of an entire biological system. Imagine two people sitting a short distance apart on the same warm summer evening. The air temperature is nearly identical around them. Both are breathing, giving off heat, and carrying exposed skin. Yet one person receives far more mosquito attention. The difference begins before the mosquito reaches either body. Carbon dioxide leaving the lungs provides a broad signal that a living vertebrate is nearby. Once that signal activates host-seeking, visual information can help the mosquito orient toward objects that stand out from their surroundings. At closer range, body-associated warmth adds more precise information. Research with Aedes aegypti has shown that thermal infrared from skin temperature surfaces can contribute to this final approach when combined with other host cues. The mosquito eventually enters the chemical cloud surrounding each person. Here, individual differences become especially important. Human skin constantly releases compounds from sweat glands, sebaceous glands, and normal chemical reactions at the surface. Microorganisms living on the skin modify some of those secretions and create additional volatile molecules that enter the surrounding air. The resulting odor mixture is different for every person. Experiments published in 2022 showed that some people remained consistently more attractive to Aedes aegypti across repeated testing. Highly attractive participants tended to carry larger amounts of several carboxylic acids on their skin. Those acids became one of the strongest identified chemical links to persistent mosquito attraction. Genetics appears capable of influencing some of this variation. Twin studies have found greater similarity in mosquito attractiveness between identical twins than between fraternal twins. Inherited biology can affect metabolism, skin secretions, immune activity, and the environment supporting skin microbes. Daily physiology adds temporary changes on top of that longer-lasting baseline. Exercise can increase carbon dioxide output, skin temperature, and sweating. Hot weather changes the amount of moisture reaching the skin. Pregnancy can alter metabolism, breathing, circulation, and body temperature for months. The mosquito therefore encounters a moving combination of stable and temporary signals. Blood type does not need to be known before the approach. The mosquito has already made most of its navigational decisions before its mouthparts ever reach blood beneath the skin. The insect's own biology matters just as much. An Aedes aegypti female adapted to feeding around humans interprets odor differently from a mosquito, whose usual hosts are birds or other mammals. Her reproductive state changes behavior as well. A female that recently completed a full-blood meal commonly stops searching for hosts while digestion and egg development proceed. After those eggs mature, her sensory priorities shift toward finding a suitable place to lay them. Human history has helped shape this relationship. Permanent settlements provided dense groups of predictable hosts. Stored water supplied breeding habitat through dry seasons. Over many generations, some Aedes aegypti populations became increasingly specialized for living beside people and finding human scent. The same precision that makes those mosquitoes effective host seekers has now given researchers specific systems to study. Repellents can interfere with successful host contact. Traps can imitate combinations of mosquito cues. Population control programs can target reproduction or aquatic development. Understanding how mosquitoes interpret human odor can guide future approaches as the sensory system becomes clearer. So when mosquitoes seem to choose one person over another, the choice emerges from an interaction between two living systems. Human physiology shapes breath, heat, and skin chemistry. Microbes alter the odor leaving that skin. The mosquito brings its own species-specific senses, reproductive condition, and evolutionary history to the encounter. For people who consistently attract Aedes aegypti, current evidence points especially strongly toward persistent differences in skin odor chemistry, including higher amounts of certain carboxylic acids. A female that follows those signals may eventually obtain the nutrients needed to mature another batch of eggs. Days later, she can leave those eggs beside water. They hatch into aquatic larvae, pass through 4 growth stages and a pupa, then emerge as new flying adults. Some of the new females will eventually begin searching through the air themselves, responding to breath, warmth, and the chemical signatures rising quietly from human skin.